Dear students, in this class I am going to explain about central dogma. If I will say it in one line, what is central dogma? It is the one-way flow of genetic information in a cell. That is how the genetic information flows within a cell. This is called as central dogma. DNA is the master copy of the genetic information because it stores the genetic information, and it is the storehouse of the genetic information. You have read this before. Now this information, how it is stored? This information in DNA is in the form of genetic codes. That is, within the DNA, the information, that is the genetic information, is stored in the form of genetic codes. Now all cell expresses its genetic information by synthesizing specific types of proteins. This expression is done by the synthesis of specific types of proteins, that is the polypeptide chains, an unbranched chain of amino acid that are linked together by polypeptide bonds. This I had explained in the class 11th. Now what this polypeptide chain is? Now the genetic information from DNA is transferred to single stranded mRNA. That is the genetic information which is stored in the DNA. It is transferred to single stranded mRNA, and this process is called as transcription. This process is called as transcription. That is the transfer of the genetic information from the DNA to the single stranded mRNA. Now mRNA from the nucleus now goes to the cytoplasm, like here DNA, then after that this information is transcribed to mRNA. Now mRNA from the nucleus, it will go to the cytoplasm. Here you can mention it as the cytoplasm. Okay, here this is the cytoplasm. Now what will happen after that? That the mRNA from the nucleus goes to the cytoplasm and provides the genetic information to the ribosome. This genetic information is provided to the ribosomes. You have read about the function of tRNA also, which now synthesizes the specific protein. Now these ribosomes, after the translation process, will synthesize the polypeptide, that is the protein. So from the nucleus, that is the DNA, it is the storehouse of genetic information. This genetic information is transcribed to mRNA, and the mRNA will now move to cytoplasm. It transfers this information to the ribosomes, which translates the information coded from the DNA into the protein. This is the gist or the summary which is involving the main stages in the protein synthesis. Now this was proposed by the Crick, and it shows unidirectional flow from DNA to RNA and then to protein. Unidirectional flow from DNA to RNA and then to protein. Then after that, this can be summarized as that DNA, then after that replication will take place. Now this DNA, after the process of transcription, this information will be provided to the RNA, then RNA will translate the information into the protein. This is the gist, that DNA replication, it will give rise to the DNA copies, then after that transcription will take place, then from the this information will be transcribed to RNA, and RNA will translate it to the protein. This is the central dogma. Again I am recapitulating the process. That is the one-way flow of genetic information in the cell is the central dogma. DNA is called the master copy of genetic information because it stores all the genetic information, and this information is in the form of genetic codes. All cell expresses its genetic information by synthesizing specific types of protein. How the cell expresses? By synthesizing specific types of proteins. which are the polypeptide in the form of polypeptide chains, and unbranched chain of amino acid that are linked together by which bond? Peptide bond. Now the genetic information from DNA is transferred to single stranded mRNA, and this is the transcription process, and mRNA from the nucleus goes to the cytoplasm and then provides the genetic information to the ribosomes, which after the process of translation synthesizes the protein, that is the polypeptide chain. So as this is the unidirectional flow, that is the DNA, then replication, then DNA, then transcription, then RNA, then translation, and then there is a synthesis of protein. But in some cases, like in retroviruses, all the viruses do not follow this process, that is the reverse transcription takes place. This was given by Temin and Baltimore, and that is why it is called as Teminism. Like in certain viruses which are called as retroviruses, this was observed. For example, the RNA tumor viruses and HIV viruses, they are the retroviruses. That is the reverse transcription process takes place, that is the reverse of transcription process, in which the viruses can synthesize DNA from their RNA. Viruses can synthesize DNA from their RNA. Viruses can synthesize, again I am repeating, DNA from their RNA, and that is why this is called as reverse transcription. Okay, here there is a synthesis of this, in this process transcription, the DNA and then RNA. Here there is a RNA and then DNA. Now what happens, that viruses can synthesize their DNA from their RNA, which is their hereditary material. Now DNA then transfers the information of mRNA that further leads to protein synthesis. This DNA leads to the protein synthesis as the DNA transfers this information to mRNA, and then after that, by the process of translation, the protein synthesis takes place. Now the enzyme required is RNA dependent DNA polymerase. Please remember the enzyme, RNA dependent DNA polymerase, which is also called as reverse transcriptase, and this is a characteristics of retrovirus only, because other viruses do not follow this process. Please remember, it is a question of competition, that this is the characteristic of retrovirus only, other viruses do not follow this. For example, RNA tumor viruses and HIV viruses. Now they have the genes in the form of single stranded RNA. They have the genes in the form of single stranded RNA, which are meant for coding the reverse transcriptase, and ultimately this enzyme that causes the synthesis of double stranded DNA molecule from the single stranded RNA, and ultimately once the DNA is formed, it leads to the formation of mRNA, which finally synthesizes the protein. So here it is called as reverse transcription because it is the reverse of transcription. That is, the information which is coded in the DNA is transcribed into RNA in the case of transcription. But here what is happening? Opposite is happening. Then that is why it is called as reverse transcription. It is also called Teminism because it was given by the Temin. Now and the Baltimore. Like in certain viruses which are called retroviruses, this phenomena was observed, and this is the reverse of transcription. So reverse transcription process, in which what happens, that virus synthesizes DNA from their RNA. Virus synthesizes DNA from their RNA, which is their hereditary material. Now the DNA then transfers the information to the mRNA that further leads to the formation of protein synthesis. Enzyme required here is RNA dependent DNA polymerase and or the reverse transcriptase, and this is a characteristic of retroviruses. All the viruses do not follow this. For example, RNA tumor viruses and HIV viruses, they have the genes, because they have the genes in the form of single stranded RNA which are meant for coding the reverse transcriptase enzyme. This question is asked that why it happens from the single stranded RNA template, that is they have the genes in the form of single stranded RNA, they are meant for coding the reverse transcriptase enzyme that causes the synthesis of double stranded DNA molecule from single stranded RNA template, and once the DNA is formed, it leads to the formation of mRNA which finally synthesizes the viral protein. So children, please go through with this central dogma, and if any problem is there, you just ask your problem to me, draw the diagram, and properly you just go through the concept, because this is an important concept which is given in your book. What is the transcription process? I will be taking it tomorrow. Thank you.